welcome back to my channel. This is Biopulse by Chandrika. Today is day 8 and then we are discussing a very important chapter of your syllabus, that is photosynthesis in higher plants. Have you ever wondered how a tiny green leaf can produce food for entire plant? And it also support life on earth? Yes, this is incredible process, isn't it? Yes, this process is called photosynthesis and it is one of the most important topic for the NEET bio. So you must be focused for this chapter. In this chapter, we will break down how plants convert simple molecule like carbon dioxide and water into glucose using sunlight. From light reaction to dark reaction and from C3 to C4 pathway, we will cover everything today. So let's begin. So here comes the first question of the chapter, that is number 1. Chlorophyll is the option A, red pigment of leaf of all plants, B, blue pigment of leaf of all plants, C, green pigment of root of all plants, and then D, none of this. So here the answer is D, none of this. Second question. In an experiment where a part of leaf is enclosed in test tube containing KOH, soaked cotton and exposed to light, will option A, test positive for starch, option B, test negative for starch due to inability to absorb light inside the test tube, C, test negative for starch due to the inability to absorb CO2, and then D, test negative for starch due to the absence of water. Before the answer, let me explain the process what actually we are going to do here. See here, this is the plant, live plant, and then we will take a leaf and then it is exposed to the soaked cotton which is soaked in a KOH. Okay, now what is the KOH what actually the KOH do means it absorbs all the CO2. If KOH is absorbing all the CO2 means there will be no presence of CO2 for the leaf. So that what happens at this area, starch won't be present. So only if I take only one leaf, this area which is exposed to the air where CO2 is present, their presence of starch will be there. But the leaf which is inside where it is exposed to the KOH, there will be no starch presence. So here if KOH is there means there will be no food for you. This is a short trick you can remember. So if KOH is exposing to the leaf means that will be test negative for what? Starch. Because of what? Because of the inability to absorb the CO2. So the option C is right. Here the absence of CO2 is there, because of that what happens? There is a no starch. There is a negative result for that. So this is the experiment of leaf enclosed in test tube containing KOH. The next one, question number 3. From now onwards, early experiment portion is very important for your NEET syllabus, because you may often you can see the questions from this portion. So just focus. Okay. Question number 3. Match the experiment objective with the scientist who performed it. These are the scientist names given and then the process here given. Experiments, those made by these scientists. So here Priestley, Julius von Sachs, Jan Ingenhousz. So here Priestley is for role of air in photosynthesis, and then Julius von Sachs is for production glucose in photosynthesis, Jan Ingenhouse is for sunlight, role of sunlight in photosynthesis. So 1 is 3, 2 is 1, and then 3 is 2. 1 is 3, that is option D. This is the chart which I prepared to remember the experiments and what was the purpose of that experiment, and then who made that one, scientist name, experiment, and then purpose. So make sure just copy this one, or else you can take the screenshot also. This is the quick table from NCERT that which I made. Experiment, first experiment, hydrilla experiment. It was demonstrated by Jan Ingenhouse to show oxygen is released during the photosynthesis. Bubble experiment also, you can remember this one. And then bell jar experiment, which is responsible for the CO2 required one, and bell jar experiment was conducted by Joseph Priestley to prove what CO2 is necessary for photosynthesis. Role of light, black paper experiment done by Von Sachs to show light is necessary. Starch test, that is iodine test, which is done by again Von Sachs to prove starch is formed during the photosynthesis. And destarching of plant, Julius Von Sachs again, to remove pre-existing starch before experiment. Variegated leaf experiment again by Julius Von Sachs to prove chlorophyll is necessary. Engelmann experiment, Theodor Engelmann, to show which wavelength of light is effective, that is action spectrum was explained by Engelmann only. So just keep the name of the scientist and then their work and their experiments name. And super quick, easily memorable line is Ingenhouse O2, Priestley CO2, Sachs light, starch, and then chlorophyll, Engelmann spectrum. Okay, action spectrum. In one line only you can remember Ingenhouse O2, Priestley CO2, Von Sachs light, starch, and then chlorophyll, and then Engelmann is about action spectrum. So let's move to the next question again. Who performed a series of experiments that revealed the essential role of air on the growth of green plants, and when? Who performed this and then when, he is asking. Option A, Cornelius Van Til in 1787, B, Joseph Priestley in 1770, C, T. Engelmann in 1756, and then D, both A and B. And then here the right option is option B, Joseph Priestley. Role of air is explained by whom? Priestley. Fifth one, who discovered oxygen and when? Option A, Joseph Priestley, B, Engelmann, C, Jan Ingenhouse, D, Joseph Priestley. So here the right option is Joseph Priestley in 1774. To get confused, he gave two different years, so don't get confused there. In 1774, Joseph Priestley discovered the oxygen. Sixth one, who showed that sunlight is essential to the plant? Option A, T. W. Engelmann, option B, Joseph Priestley, C, Jan Ingenhouse, D, Cornelius Van Til. So here the right option is Jan Ingenhouse. Sunlight is essential for the plant is said by whom? Is experimentally proven by Jan Ingenhouse. Seventh question, part of plant that could release oxygen, that who said about this? A, Jan Ingenhouse, B, T. W. Engelmann, C, Joseph Priestley, D, none of this. So here the option A is right, that is Jan Ingenhouse. Match the following question. First action spectrum was by T.W. Engelmann. Action spectrum, T.W. Engelmann, just keep in mind. If among these, if you know any of the two, you can choose the correct one. Option A is 2 means A is 2, C, last one. Only with one correct guess I can guess the answer, isn't it? So first action spectrum is T.W. Engelmann, and then chlorophyll, Julius von Sack. O2 evolved from H2O is Cornelius van Neel, and cladophora is a alga. So this is the 8th question, and the correct option is D. 9th one, dash were used to detect the sites of O2 evolution. What were used to detect the sites of O2? That is bacteria, B, daisy, C, fungi, and then D, virus, and then correct option is bacteria. 10th one, Cornelius van Neel studied on dash to demonstrate that photosynthesis is essential a light dependent reaction. A, cladophora, B, purple and then green bacteria, C, red algae, D, both A and B. So here the right option is B, purple and then green bacteria. Cornelius van Neel studied on a demonstrated that photosynthesis is essential, a light dependent reaction. Purple bacteria and then green bacteria he used. That means those bacteria were exposed to the particular light of the spectrum. 11th one, dash from a suitable oxidizable compound reduces CO2 to carbohydrates. A, oxygen, B, hydrogen, C, carbon, D, both A and B. And then right option is option B, hydrogen. 12th one, cladophora is a option A, purple and then green bacteria, B, green bacteria, C, red algae, and then D, green algae. And then here the right option is D, it's a green algae. 13th one, A, O2 evolved by the green plants comes from H2O, not from the carbon dioxide. B, this was proved by using radioisotopic technique. Yes, both these statements are correct. So option D is correct. Now the questions from the concept, where does photosynthesis takes place? 14th question, where does photosynthesis takes place? Option A, green part of leaf. Option B, green part of stem. C, brown part of stem. B, both A and B. So here the right option is D, both A and B. Next, question number 15, assertion and reason question. Assertion here is, chloroplasts usually align themselves along the walls of mesophyll cells. Reason, they get optimum quantity of incident light by aligning along well. Choose the correct option. So both are, assertion and reason are correct, and reason is the correct explanation of assertions. That's what A option is correct. 16th one, chloroplast is option A, single membrane organelle. No. Option B, double membrane organelle. Correct. So here the right option is option B. 17th question, choose the incorrect statement from the following. Option A, there is no clear division of labor within chloroplast. B, chloroplast has membranous system which includes stroma as well. 3. C, membrane system is responsible for trapping the light energy. And then D, more than one of the above. So here the incorrect statement is, there is no clear division of labor within the chloroplast, but chloroplasts show a very correct labor of division. means particular light reaction will be takes place in thylakoid membrane, and then dark reaction will be takes place in stroma. Those are enzymes are responsible for such kind of these processes. So here this statement is wrong. Next, 18th one, dark reaction. Option A is not light dependent. B occurs in darkness. C is photochemical reaction, and then D is indirectly light dependent. So here the right option is D. Dark reaction is indirectly light dependent, because dark reaction will takes place only when the light reactions products are there. Next, 19th one, this is the structure of the chloroplast. Chloroplast has a double membrane system, and this is the outer and then inner membrane, and these are the grana. This one grana, and then this one is one more grana. The membrane in between two granas are called stroma lamellae. There will be a starch granules, there will be lipid droplets also, small black drops are showing, no, those all are called what? Lipid droplets. So here let's guess what it is actually. Just now I said this is grana, isn't it? If I know third one is grana, where is grana? Only B and C must be the right option. And one more option I know. Which one I know? This is lipid droplet, I know. So one is lipid droplet. So starch granule is wrong. Again, by the elimination method I can go with the right option, that is C. 20th question. Sugar is synthesized A. non-enzymatically in grana, B. non-enzymatically in stroma, C. enzymatically in grana, D. enzymatically in stroma. So here the right option is D. enzymatically in stroma, because the sugar, what is the glucose is there, no, that is the end product of dark reaction. Where the dark reaction takes place? Dark reaction takes place in stroma. That's what. 21 question. Which of the following is correct? Option A, light reaction depends on dark reaction. B, dark reaction depends on light reaction. C, both of the above. D, none of the above. So here the right option is B, because dark reaction depends on light reaction. Actually it is depend on the products of light reaction. 22nd question. If a plant is kept in dark for a long time, option A, starch will be synthesized in chloroplast. B, ATP will be synthesized in chloroplast but no starch. C, NADPH will be synthesized in chloroplast but no starch. D, none of this. So here the option none of this is correct, because if a plant is kept in dark, what happens? If there is no light, in the sense there will be no any such kind of the photosynthesis and their products, whatever it may be a ATP, it may be a NADPH, there will be no products. If there will be no any light products, then there will be no presence or else synthesis of the starch. 23. Choose the incorrect option during daytime. A, ATP will be synthesized in chloroplast by light reaction. B, NADPH will be synthesized in chloroplast by light reaction. C, starch will not be synthesized by dark reaction. And then D, none of these. So among them, which one is incorrect option he is asking. These both are correct, and then C is wrong, because starch is synthesized by dark reaction. Dark reaction product itself is the starch. How many types of pigments are involved in photosynthesis? 24th question. The color of leaf is due to option A, chlorophyll only. B, chlorophyll carotenoids only. C, chlorophyll carotenoids and xanthophylls. And then D, none of this. So here the right option is chlorophyll carotenoids and xanthophylls. 25th question. Leaf pigments are separated by, the pigments of the leaf are separated by which process? Option A, crystallization. No. Option B, gel electrophoresis. No. C, blotting. D, paper chromatography. Here the right option is paper chromatography. Match the following. Match the pigment with its color. Chlorophyll A, chlorophyll B, carotenoids and xanthophyll. Chlorophyll A shows blue and then green. B, yellow green. So one is for A and then B is for C. So right option is B. 1 is for A, 2 is for C, and then 3 carotenoid is for D, xanthophyll is for B, yellow. So here the right option is B. And then quick remember, there is a shortcut or else there is a trick which will be remembering BBY, blue bright yellow, or else you can also remember like this: A is for blue green, B is for yellow green, and carotenoids yellow orange, or else simpler than that, BBY. Okay, BBY, blue green. B in the sense bright means yellow, so yellow one. Okay na, BBY. ABC, BBY. A is for blue, B is for bright yellow, and then C is for yellow orange, yellow green, yellow orange. 27th, which is the most abundant plant pigment in the world? Most abundant plant pigment in the world is one and only the best pigment, I can say, that is the king, that is the chlorophyll A, because this is the main pigment which will absorb the light, and then this is the reaction center. Without the chlorophyll A, you can't imagine the world itself. Shortcut to remember what actually their primary pigment, accessory pigment, and then necessary pigments. A, B, C. A, B, C. That is chlorophyll A, chlorophyll B, and then chlorophyll A, B, and carotenoids. Chlorophyll A is primary pigment because it absorbs, and then it is the main reaction center, and then chlorophyll B is accessory pigment. This one is necessary, and then these two are accessory pigments. Rather than the chlorophyll, carotenoids and then xanthophylls are accessory pigments. Got it? A, B, C. Keep in mind A, B, C. Among the A, B, C, which one is primary? A, chlorophyll A is primary. Remaining B and C are accessory pigments. Only A can act, others just attract. A is going to act here, others like chlorophyll A, chlorophyll B, and then C are going to attract. Attract whom? Sunlight. Not the sunlight actually, that is the light. 28th question. Identity, correct, identify the correct one. 1, 3, and then 2. Which one is 1, which one is 2, and then which one is 3? Here the right option is option D. How you can see? See, always keep in mind there will be chlorophyll B's peak is more. Okay, at which area? Okay, now this is the blue one, chlorophyll A absorption spectrum, and then this one is chlorophyll A absorption spectrum, and then this one yellow one is carotenoids. This one is B, this one is A. Otherwise, you can remember like this: who came first, okay, they go last. Who came next, they go first, isn't it? This is very quickly you can remember. First, chlorophyll A's absorption spectrum is first, isn't it? But its absorption spectrum goes last. When the absorption spectrum of chlorophyll B you are seeing, it is next to chlorophyll A, but it is showing its low before the chlorophyll A. Okay, I think this is very easy. जो फर्स्ट आएगा वो लास्ट को चला जाएगा, जो नेक्स्ट आएगा वो फर्स्ट चला जाएगा. I hope you people are clear about this. And then the yellow will be about the carotenoids. 29th question. Maximum absorption by chlorophyll A occurs in A. Blue and then green region, B. Red and then green region, C. Blue and red region, D. Yellow and red region. So maximum absorption of the chlorophyll is at the blue and then red region. 30th one, assertion and then reason. Chlorophyll A is chief pigment associated with the photosynthesis. Exactly. And then chlorophyll maximum absorption coincides with maximum photosynthesis. So among them choose the correct option, means assertion and reasons are correct. Reason is the correct explanation of what? Photosynthesis. 31st question. Accessory pigments includes. Accessory pigments includes option A chlorophyll A, B chlorophyll B, option C both of them, and then option D none of this. So here the right option is chlorophyll B. Chlorophyll A is only the chlorophyll A is necessary pigment, where rather than that B and then carotenoid and xanthophyll are the accessory pigment. 32. Accessory pigments, option A pass on a energy to chlorophyll A, B pass on energy to primary acceptor, C use energy for photolysis of water, D more than one option is correct here. So here the right option is pass on energy to chlorophyll A. If I say, let me draw, just imagine this is the this is the site of chlorophyll A. Okay, chlorophyll A is surrounded by many of the pigment molecules. Those may be chlorophyll B, chlorophyll B, carotenoid, and then xanthophylls. Okay, whenever the light quantum is exposing to the leaf, especially the area where the absorption, who is going to be mainly absorbing the chlorophyll A? It is the reaction center actually, but remaining whatever the wavelength of the light is there, no, that is not going to waste there. They are absorbed by the accessory pigments which are around the chlorophyll A, and they will pass on the energy to the next, and then again they will be passing on, and finally they are handing over their energy to whom? To chlorophyll A. So this is what actually passing of the energy to whom? Chlorophyll A. That's what the right option is, option A. Here absorption is takes place at the blue and red region. That's what here I wrote. Blue and red are for fed and green is dead. Why green is dead? Due to where at the region of green there is no absorption of the light. When absorption of light is not there, green is going to be reflected back. That's what our plant looks green in color. But what our plant is going to be accepting, it is accepting or else it is absorbing the light in two regions, that is the blue one and then red one. That's what the blue color and then red color of the light are fed and then green is dead. That's what our plant looks which color? Green in color. Next question 33. Advantage of accessory pigment. Advantage of Advantages of accessory pigments include option A. They help by photolyzing the water. B. They protect chlorophyll A from photooxidation. Exactly. C. They enable narrower range of wavelength of incoming light to be used for photosynthesis. And then D. Both A and B. So here the right option is B. They protect chlorophyll A from photooxidation. Just now I show you the diagram, no? Along with the passing of the energy to the chlorophyll A, whenever these pigments are surrounding the central chlorophyll A, no, they will protect the pigment from photooxidation. Next, now from light reaction. What is actually light reaction? 34th question. Light reaction is also known as option A. Photochemical phase, option B. Biosynthetic phase, option C. Both of this, and then D. None of this. So here the right option is option A. Photochemical reaction. Light reaction. Light means photo and then reaction where it is going to take place a chemical phase. That's what it is called, what? Photochemical phase. 35. Choose correct order of events in light reaction. Order of events in light reaction. First ATP and NADPH formation. No, it will be last. Why? Because first what happens in light reaction? Absorption happens. Light absorption. Which one is fourth one? Where is fourth one? 2 and then B and C is having fourth one. Next, oxygen, water splitting and oxygen release. After the absorption of light, there will be splitting of water. After splitting of water only there will be releasing of the oxygen is there, no? After that what happens? There will be a end product who are said to be a ATP and then NADPH formation. So one must be in the last and then fourth must be in the first. 1 is last, 4 is first. So option C is correct here. LHC stands for A. Late harvesting complex, B. Light harvesting complex, C. Light hanging complex, D. Late hanging complex. Late word. By saying late word only I am getting laugh. Here the right option is light harvesting complex. What is light harvesting complex means? Let me show you. This is chlorophyll A and then other pigments surrounding the chlorophyll A, isn't it? Whenever the photon is exposing, sunlight is exposing, whatever the absorbed energy is there, no, it is going to be passed to the adjacent molecule or else adjacent pigment, and then finally it is going to be handing over or else passing to the chlorophyll A. And whatever this harvesting is there, no, means absorbing of the light from all the pigments in different wavelength, that harvesting, and then finally it is passing to the chlorophyll A. That complex, this kind of complex is called what? Light harvesting complex. Best example for this, class monitor, isn't it? Class monitor will do? If I ask her or him to collect all the notes, she or he will collect all the notes, and then they will hand over that notes to whom? To the teacher. They will keep that notes to the bench. Or else one more example I can give. In examination, invigilator, sometimes invigilator, what he or she will do, she will give bundle of papers, and then she will just ask them to pass at the beginning of the examination. After the end of the examination, what she will ask to collect. What the students will do, students will pass their answer sheet to next to them, and so that, like that, what happens, all the students will take, they will receive their friends answer sheet also, and then finally it is given to the madam. So here madam is chlorophyll A, and then all the students are comes under light harvesting complex. Got it now? That's good. 37th one, the naming of PS1 and PS2 was based on A. Their discovery order, B. Their functioning sequence, C. The scientist who named it, and then D. The component of the photosynthesis. Here the right option is their discovery order. First discovered by photosystem 1, photosystem 2, and then photosystem 1 next discovered. 38th one, which of the following is correct? Option A, photosystem 1 is called P800. B, photosystem 2 is called P680. C, both A and B. D, none of this. Here the right option is B. PS2 is called P680, PS1 is called P700, not 800. 39. Reaction center is formed by A. Only one chlorophyll A molecule, B. A few chlorophyll A molecule, C. One chlorophyll A and a few accessory, D. A few chlorophyll A and a few accessory pigments. Here the right option is A. I said reaction center is formed only with one chlorophyll A molecule because it is the master. It's the king there. It's his territory. No one is allowed there. 40th one. Choose the incorrect statement. Option A. Antenna is a light harvesting system. B. Contains accessory pigments. C. Doesn't include reaction center, and then D, none of this. Here the right incorrect statement, all are correct statement only, that's what the option is, none of this. Electron transport. 41. When the light energy is absorbed by PS2, it is option A, converted to mechanical energy, B, used to excite electron, C, used to change configuration of the Rubisco, D, both A and C. Here the right option is B, used to excited electrons. The movement of 42 question, the movement of excited electrons in non-cyclic photophosphorylation. A, uphill in terms of reduction potential scale, B, downhill in terms of reduction potential scale, C, uphill and downhill in terms of oxidation reduction potential scale, D, both A and C. Here the right option is C. 43. Electrons excited from PS2 A get used up by the first electron acceptor. B get used up in the middle of its ETS pathway to PS1. C get used onto pigments of PS1. D get partially used up in ETS and the rest is passed to PS1. So here the right option is get passed on to the pigments of PS1. Excited electron is going to be passed to the PS1. Let me show you. If I am saying this is PS2, okay, and here there is one more light harvesting system, that is called what? PS1. There is a central master molecule which is called chlorophyll A. Here what happens, whatever the light harvesting system is absorbing light, electrons are going to be excited. The excited electron is excited and then it is accepted with the first electron acceptor. Then next what happens, it is going to be passed to whom? To PS1. The next it is processing to the next one. Lastly it is used with the products, that is ATP and then NADPH. So here the right option is C. 44. The electrons passed on by PS1 to electron acceptor are A. The ones that were transferred to PS1 from PS2. B. Electrons from water splitted. C. Electrons excited when PS1 absorbs light, and D. All of this. So here the right option is electrons excited when PS1 absorbs light. Again, just now I said no. Here whenever the excited electron is excited, again it is going to be in a downwards. Again if it is receiving the light, again electron will be exerted. So here the right option is C. 45th one. Electrons from PS1 move downhill to a molecule of energy rich. To whom it is going to be giving that energy? Option A NADPH plus, option B NADP plus, option C FAD plus, option D GTP. Here the right option is option A. NADP plus is receiving that energy to form a NADP plus H plus. 46. The Z scheme is named so because A. It was discovered by a scientist with Z as initial letter of name. No. B. The carrier of a ETS present in thylakoid member are in Z shape. C. It forms a Z shape when the carriers of ETS are arranged in a sequence on a redox potential scale. D. Both A and C. Here the right option is C. When I say this is PS2 and this one is PS1, okay, photosystem 2, photosystem 1. If light is or if the electron is excited, it moves uphill, isn't it? And after the energy, then after the excitement, when it is accepted with the primary acceptor, again it is going to move downwards. This is downward. Again if it is receiving the light energy, then again the electron is going to upwards. Up, down, up. If you see, this is like a Z. That's what it is given the name Z scheme. It forms Z shape when the carriers of ETS are arranged in a sequence on redox potential scale. 47. Identify the correct site for 1. That is water splitting, 2. NADPH, plus reduction, and then 3 ATP synthesis. Here they are giving a numbers. This is 1, this is 2, and then this is 3. What this one we must identify. What is 1? What 1 is electron transport system, and then 2 is water splitting. 1 is electron transport system, which is having actually a ATP synthesis, and then 2 is water splitting, and then 3 is reduction. So 1 ATP synthetase means 1, it must have 3. 3 has 2 options, so A and B are wrong. Let us check second one. 2 is water splitting. 2 must have 1, so option D is correct. Yeah. And then 3 is NADP plus reduction, so 2 is here. So 47th one, right option is D. Now splitting of water. 48th one, splitting of water is important. A, for the oxygen formation. B, for the electron released. C, for the hydroxide ion released. D, for H2 release. So here the right option is option B, for the electron release. 49th one, water splitting is associated with option A, PS1 located on inner side of the thylakoid membrane. B, PS2 located on inner side of the thylakoid membrane. C, PS1 located on outer stroma lamellae. D, PS2 located on outer stroma lamellae membrane. So here the right option is option B. Always the water splitting complex is with PS2, keep in mind. 50th one, the O2 is released in A, lumen of thylakoid, B, outer side of thylakoid, C, stroma, D, cytoplasm. Here the right option is option A, lumen of thylakoid. See how actually it will be. If I show you thylakoid membrane, this is thylakoid. Thylakoid is also having 2 membrane system. Okay, and this is the lumen. Somewhat imagine here is PS2 and then PS1 is here. PS2 always have the complex of water splitting. So after the splitting of water, what happens? Whatever the oxygen is there, no, that oxygen is released where? In lumen itself. So here the right option is lumen of thylakoid. Are you getting which diagram actually I drawn? Chloroplast, inside the chloroplast there will be a like stacks of thylakoid, that is called grana. In that grana only one thylakoid diagram I drawn here. 51, protons are released. Option A, in lumen of thylakoid. Option B, outer side of thylakoid. C, stroma. D, cytoplasm. Here the answer is again inside the lumen of the thylakoid. Due to that, what happens? Due to that only proton gradient will increase there, due to which ATP production will be there. Cyclic and non-cyclic photophosphorylation. 52. The process of ATP synthesis in cells is or are option A, photophosphorylation, B, oxidative phosphorylation, C, phosphosynthesis, D, both A and B. Here the right option is option D, both A and B. Cyclic and non-cyclic photophosphorylation, in that process of ATP synthesis in cells is by both photophosphorylation and oxidative phosphorylation. 53rd question. The order of working of the two photosystem is option A PS1 first then next PS2, option B PS2 first then next PS1, C any of this depending upon location, D none of this. Here the right option is PS1 will be first then next PS2. 54. Non-cyclic photophosphorylation involves A PS1, B PS2, both PS1 and 2, D none of this. Only enzymes in stroma. No. So here the right option is both PS1 and PS2 are used in non-cyclic. In cyclic only PS1 is going to use. 52. End product of Z scheme is option A ATP, B glucose, C NADP plus H plus, and then D both A and C. Here the right option is option D, both A and C. I will show you how. See, this is PS1, this is PS2, and this is PS1. Okay, whenever the light energy or else light wavelength in the form of quantum is absorbed by the chlorophyll A, electron is going to be excited and then it is accepted by the primary acceptor. Then next it is following to the ET, electron transport system. Here in the electron transport system there will be production of what? ATP. After that electron will pass to PS1. Again by receiving the light energy, again they are going to be excited. After that what happens? Those electrons are passing to the final acceptor of that is array NADP plus. Okay, NADP plus will convert in the after the receiving the electron, it will form NADH plus H plus. So here the right option is products will be ATP and NADH plus H plus. Next, 56. Cyclic photophosphorylation involves PS1 only, B PS2 only, C both PS1 and PS2, D none of this. It involves only PS1. 57. Cyclic phosphorylation ends in the formation of A ATP only, B glucose only, C NADPH plus H plus only, D both A and C. So here the right option is option A. Let me show you. So in cyclic what happens? There will be only involvement of PS1. Electron is going to be excited. Okay, excited electron is transferred to the electron transport system. Just imagine this is electron transport system. Again that electron is moving back to the site. That's what it is called cycle. The excited electron is again moving back to the same area. Okay, during that what happens? Only production of ATP will be there. 58th one. A possible location of cyclic photophosphorylation under full light is condition. Option A stroma, B stroma lamellae, C cristae, D outer membrane of chlorophyll. Here the right option is stroma lamellae. 59. Assertion and reason. Cyclic photophosphorylation occurs in stroma. Reason: stroma membrane lacks PS2 and NADP reductase. Choose the correct answer. Here the correct answer is B, where both assertion and reasons are correct, but the reason is not correct explanation for assertion. Now, chemiosmotic hypothesis, one of the best hypothesis I can say. 60th question, ATP synthesis is linked to dash graduate across a membrane in dash. ATP synthesis is linked with dash and then across a membrane in a dash. So here first one, electron electron proton proton. Second one, they link to a electron gradient across the membrane in photosynthesis and respiration. Photosynthesis only, photosynthesis and respiration, photosynthesis but not the respiration. Here the right option is option C, proton and photosynthesis and respiration, because ATP synthetase enzyme is used for both in photosynthesis and respiration process. Next, 61st question, which of the following statement is true? A, a proton accumulates towards outer side of the membrane in photosynthesis. B, the protons accumulate towards inner side of thylakoid in respiration. C, the protons accumulate towards outer side of the membrane of the respiratory thylakoid. And then D, none of this. Here the statement D, because all are wrong only. 62th question, the proton gradient may be formed in photosynthesis is due to A, splitting of water, B, reduction of NAD plus, C, both A and B, D, none of this. Here the right option is splitting of water. Proton gradient, the main source of proton gradient is by splitting of the water only. I will show you how. See, whenever the PS2 is working, no, for the PS2 there will be a complex called as water splitting complex. It will split the water, just a minute, H2O into what? After splitting one molecule of water, we will be getting 4 protons okay, and 2 electrons, and the byproduct, that is oxygen, what we are breathing now. Okay, whenever the water is going to split, the proton gradient will be increasing. So here the right option is splitting of water. Next one, assertion and reason type of question. 63, assertion is protons move through photosystem, protons are transported across membrane. Reason, primary acceptor of electron is an hydrogen carrier. So option A, option B, option C, option D. Here the right option is option A. Both are correct and then explanation is also correct. 64, which of these is an hydrogen carrier? A, PS2, B, pyridoxine, C, plastocyanin, D, plastoquinone. Here the right option is D, plastoquinone. Keep in mind, always the primary acceptor of the electron by the PS2 is plastoquinone, that is PQ. Next, 65, NADP reductase enzyme is located on dash of thylakoid membrane. stroma side, outer side. B. lumen side, that is outer side. Stroma side, that is inner side. Lumen side, that is inner side. Here the right option is A. stroma side, outer side. NADP reductase, stroma side will be there. Means let me show you. If I draw this as a thylakoid membrane, this is the outer one and then this one will be inner one, not like this. Please excuse me for this. This is PS2 and then this one is PS1. Okay, whatever the electron is excited, they will be moved like this next to the PS1 and next last here. Here somewhat what happens, there will be a NADP reductase which is the enzyme to form a NADP plus H plus. Okay, that's what the answer, stromal side. '66. The proton gradient is also formed due to A. reduction of NAD plus, B. reduction of NAD plus, C. reduction of both NAD plus and NADP plus, and then D. reduction of FAD plus. Here the right option is B. '67. Which of the following is true? A. ATP and NADP H plus both are formed towards stroma. B. ATP and NADP H plus both are formed in lumen of thylakoid. C. ATP is formed in lumen while NADP H plus is formed towards the stroma. D. NADP H plus formed in lumen while the ATP is formed in stroma. This is wrong actually. So here the right option is A. '68. Identify the correct label. Here they given 1, 2 and then 3. Among them, if you know any one, you can easily identify. So here 1 is cytochrome B6F, and then last one is plastocyanin. This is plastoquinone, which is the first acceptor. So right option is option D. '69. Identify the correct one. 2 and then 3. A. high electron gradient. First one is high electron gradient. Second one low electron gradient. No, here the electron gradient is more. This is the here it is low and then here it is high. So here the right option is 1 is low electron gradient and then here second one is 2 high electron gradient. 70th one. Statement A. The accumulation of proton in stroma cause decrease in pH of stroma. B. Accumulation of proton in lumen cause formation of potential gradient across thylakoid membrane. Select the correct option. Here the correct option is option B. B is correct, A is incorrect. 71. ATP formation occurs upon A. build-up of potential gradient, B. breakdown of potential gradient, C. both of these, and then D. none of this. Here the right option is option B. After the breaking down of the potential gradient, the formation of ATP takes place. 72. The transmembrane channel in ATP synthesis enzyme is formed by A. CFO, B. CF1, C. CF2, and then D. both A and B. Here the right option is A. CFO. 73. The transmembrane channel allows across membrane of ATP synthesis. Option A, osmosis of protons. Option B, simple diffusion of protons. Option C, facilitate diffusion of electron. D, facilitate diffusion of proton. Here the right option is facilitate diffusion of protons. 74th question. The ATP synthesis has two parts, 1 and then 2. This is the one, this one is 1 and then this one is 2. What is 1 is called and then what is 2 is called? Here the right option is 1 is called CFO and then 2 is called CF1. CF0 or CFO, CF0 is embedded in the membrane, that is thylakoid membrane. This is thylakoid membrane, and here CF1 is outside towards the stroma. 75. CFO is or CF0 A, embedded in chloroplast membrane. B, protruding on outer surface of chloroplast. C, protruding on inner surface of the chloroplast membrane. D, none of this. Here the right option is option A. Next, 76. CF1 is option A, embedded in chloroplast membrane. B, protruding on outer surface of chloroplast membrane. C, protruding on inner surface of chloroplast. D, none of this. Here the right option is option B. Actually, rather than chloroplast membrane, the thylakoid membrane that must be. 77. Conformational change in dash makes ATP in CFO or CF0. B, CF1. C, both. D, none. CF1. Conformational change will occur in CF1 part. 78. For creating proton gradient across the thylakoid membrane, A, energy is used. B, no energy is used. C, energy is released. D, none of this. Here the right option is option A, energy is used. 79. The end product of light reaction are A, stored till dark reaction takes place at right. B, immediately used up in the next round of light reaction. C, transferred to the stroma from the lumen to be used in biosynthetic reaction occurring in the stroma. So here the D, none of this. Next question. Where are ATP and NADPH used? 80th question. The product of light reaction are A, ATP only. B, ATP and NADPH. C, ATP, NADPH and O2. D, NADPH only. So here the right option is ATP, NADPH and O2. 81. O2 is A, used up in dark reaction in stroma. No. B, used up in dark reaction in lumen thylakoid. C, diffused out of chloroplast. More than one option is correct. Here the right option is option C. It is diffused out of the chloroplast, that we can, what actually we are going to breathe. Whatever the oxygen we are breathing, no, that is diffused out by the chloroplast, the byproduct of the photosynthesis. 82. Statement A, biosynthetic reaction is independent of direct presence of light. B, biosynthesis process continues for some time after the light becomes unavailable and then stops. No. Here both A and B are correct. 83. Kelvin discovered that first CO2 fixation product is A, 3 carbon organic acid. Yes, that one is. B, 4 carbon organic acid. C, 5 carbon organic acid. D, 6 carbon organic acid. Here the right option is option A. That's what Kelvin cycle is also said, that said as C3 cycle. 83. To discover the first CO2 fixation product, Kelvin worked on using option A algae radioactive C12, fungi radioactive C12, algae radioactive C14, fungi radioactive C14. So here the right option is C radioactive 14 algae. 85. The first product of CO2 fixation was identified to be in Kelvin cycle A. PGA, B. RUBP, C. Citric acid, D. OAA. That is option A, PGA, phosphoglyceric acid. 86. In C4 pathway, first CO2 fixation product is same options as option A, PGA, B. RUBP, C. Citric acid, D. OAA. So here the first stable product fixation product is OAA in C4 and C3 it is PGA. 87. OAA and PGA stands for option A 3-phosphoglyceric acid oxalate acetic acid respectively, B 3-phospheptidoglutaric acid, no, phosphoglutamic acid, no. So here the right option none of this. This is oxaloacetic acid and this one is phosphoglyceric acid. Phosphoglyceric acid is right but the oxaloacetic acid is wrong. Oxaloacetic acid, phosphoglyceric acid and then oxaloacetic acid. The primary acceptor of CO2. 88. For 3-carbon compound to be formed after the CO2 fixation, the acceptor molecule is of A 2-carbon, B 3-carbon, C 4-carbon, D 5-carbon. Here the right option is D 5-carbon, that is RUBP. Next, 89. RUBP stands for A. Ribulose bisphosphate carboxylase oxygenase, B. Ribose bisphosphate carboxylase oxygenase, C. Ribulose bisphosphate, D. Ribulose bisphosphate carboxylase oxygenase. Here the right option is option C, ribulose bisphosphate. Next, from the Kelvin cycle, 90th question. The Kelvin cycle starts with dash ends with. The Kelvin cycle starts with whom and then ends with whom? A. Rubisco, regeneration of Rubisco, no. B. RUBP, regeneration of Rubisco, no. C. RUBP and then regeneration of RUBP. This one is the correct one. 91. Kelvin cycle A occurs in C3 plants only, B occurs in C4 plants only, C occurs in both C3 and C4, D none of this. Here the right option is option C. 92. This is what actually Kelvin cycle is, which is having 3 main steps: carboxylation. Yes, this is reduction and then regeneration. So here the right option is 1 is carboxylation, 2 is reduction and then 3 is regeneration. So right option is option C. 93. The most crucial step of Kelvin cycle is carbonation, B carboxylation, C reduction and then D regeneration. Here the right option is B carboxylation. 94. Carboxylation is catalyzed by the enzyme A RUBP, B PEP, C NADPH and then D none of this. Here the right option is D none of this, because carboxylation is catalyzed by the enzyme. They are not present here, that is the enzyme Rubisco. 95. Rubisco has the activity of A. Carbonation, B. Oxygenation, C. Oxidation. None of this. Oxygenation is right. 96. Reduction involves use of dash molecules of ATP for fixed CO2 reduction. Reduction involves use of dash molecules of ATP to fix CO2. Here 1, 2, 3 and then 4. The answer is 2. 97. Reduction involves use of molecules of NADP to reduce fixed CO2. Here the right option is again 2 molecules of NADPH. 98. For formation of one glucose molecule, how many turns of Calvin cycle is needed? 3, 1, 2 and 6. Here the right option is 6. 99. Regeneration of dash takes place at expense of dash ATP and dash NADPH. Regeneration of Rubisco takes place at expense of 1 ATP and 0 NADPH. 100. The centuryth question. Statement A. In CO2 fixation cycle, the molecule of ATP used is more than NADP used. Statement B. To meet the difference in number of ATP and then NADP used in dark reaction, cyclic phosphorylation takes place. Choose the correct option. Here the correct option is A and B are correct, and then A explains the B. 101. For formation of one glucose, how many molecules of ATP are required by C3 cycle to form a one glucose? It requires 18 molecules of ATP. 102. For one glucose formation, how many NAD molecules are needed by C3 pathway? It is needed 12. ATP 18s and then 12 NADPs. In C3, keep in mind, C3 pathway requires, for one formation of the one glucose, it requires 18 ATPs and then 12 NAD molecules. From now, C4 pathway questions. 103. C4 plants are adaptation of the plants to A. Wet regions, B. Polar regions, C. Dry tropics and then D. Moist rainforest. So here the right option is dry tropics. 104 question. 1. C4 plants lack Calvin cycle. 2. C4 plants lack photorespiration. 3. C4 plants have more productivity than the C3 plants. And then 4. C4 plants cannot tolerate higher temperature. How many of the above statements are incorrect? Incorrect they are asking. Here 2 are incorrect. 105. First CO2 fixation product in C4 cycle is RBP, PEP, OAA, malate. First CO2 fixation product is OAA. There in C3 it is PGA, in C4 it is OAA. 106. Bundle sheath cells are present in dash around the dash. Option A. C4 plants vascular bundle, B. C3 plants vascular bundle, C. Both of these, none of this. Here the right option is option A. C4 plants vascular bundles. 107. Leaves with bundle sheath called are said to show A. Crant anatomy, B. Cran's anatomy, C. Kent anatomy, D. Kren's anatomy. Here the right option is option B. Cran's anatomy. Next question. 108. Bundle sheath cells allow gaseous exchange, have intracellular space, have large number of chloroplasts, D. All of this. Here the right option is C. Have large number of chloroplasts. 109. Example of C4 plant is A. Rice, B. Maize, C. Soya bean, D. Both A and C. Here the right option is B. Maize. 110. Primary CO2 acceptor in C4 plant is A. 3 carbon molecule RUBP, B. 3 carbon molecule PEP, C. 4 carbon molecule PEP, D. 4 carbon molecule OAA. Here the primary acceptor is 3 carbon molecule PEP. Phosphoenol pyruvate. 111. Enzyme responsible for primary CO2 fixation in C4 plants is A. Rubisco. Rubisco is responsible in C3 plants. C3 cycle. Okay. B. Pepcase. C. Oxaloacetate. D. Phenolase. Here the right option is B. 112. Which of the following is true? Option A. C4 plants lack Rubisco. B. Mesophyll cells of C4 plants lack Rubisco. C. Bundle sheath cells of C4 plants lack Rubisco. D. C3 plants lack Rubisco. Which of the following is true? Here the right option is option B. Mesophyll cells of C4 plant lack what? Rubisco. If I show you the C4 plant mesophyll cell and then bundle sheath, this is the mesophyll cell and then this will be the bundle sheath cell. This is mesophyll and this is bundle sheath. Here which enzyme is present? Here PEP is present. Pepcase is present here. Pepcase is an enzyme. Okay. Here Rubisco is present. Rubisco is present. In mesophyll cells, Rubisco is absent. In bundle sheath cell, pepcase is absent. 113. Primary CO2 fixation occurs in C4 plants in A. Bundle sheath cells, B. Mesophyll cells, C. Any of the above, D. None of the above. So here the right option is mesophyll cells. 114. CO2 fixation in C4 plants occurs in bundle sheath cells, just now only, no? The primary acceptor. Okay. CO2 fixation in C4 plants occurs in bundle sheath cell. Fixation he is asking. Don't get confused like me. Just now I got confused. Here primary CO2 acceptor fixation he is asking. And here CO2 fixation in C4 plants occurs. So here the right option is option C, both. Here in bundle sheath for C3 cycle CO2 fixation will be there. In mesophyll for the primary acceptor CO2 fixation will be occur. 115. OAA forms other 4 carbon acid which are transported. They are A. Malic acid and oxalic acid, B. Malic acid and aspartic acid, C. Succinic acid and aspartic acid, D. Succinic acid and then glutamic acid. Here the right option is option B, malic acid and then aspartic acid. 116. This figure shows Krebs cycle, Calvin cycle, Hatch and then Slack cycle, EMP pathway. Here the right option is option C, Hatch and then Slack pathway. This is the Hatch and Slack pathway. This is mesophyll cell and then this is bundle sheath cell. Here the Rubisco is absent. Here the Rubisco is present. Here the Rubisco is absent. Here pepcase is present, but here pepcase is absent. 117. In C4 pathway, Rubisco is option A, absent, B, present in mesophyll cells, no, present in bundle sheath cells, correct. 118. Peptidase enzyme is A. Absent in mesophyll cells, no. Present in bundle sheath cells, no. Both A and B, D. None of this. Here the right option is none of this. 119. Calvin cycle takes place in dash in all C3 plants in dash in all C4 plants. Mesophyll cells. Calvin cycle takes place in dash in all C3 plants in dash in all C4 plants. 2. Bundle sheath, it must be mesophyll cells in all C3 plants. So right option is option C. 120. Identify the correct label. First one is fixation. So here the right option is regeneration, transport and fixation. This one is 1. This is this one is 2 and then this one is 3. Identify the A and B. Here what is A and then what is B? This is A and this one is B. This is mesophyll cell and then this one is bundle sheath cell. Very easy. Now next about the photorespiration. 122. Read the following statements. Statement A. Ribulose bisphosphate is the most abundant enzyme in the world. Correct. Statement B. Photorespiration doesn't occur in C4 plants. So here the right option is option C. Both are correct. 123. Rubisco has dash when CO2 and then O2 is equal. A. Greater affinity for CO2 than the O2. This one is itself is right. Rubisco always has a much more affinity towards the CO2. 124. In C3 plants, option A, some O2 binds to Rubisco and O2 fixation increases. B. No O2 binds to Rubisco. C. Some O2 binds to Rubisco and CO2 fixation decreases. And D. Only O2 binds to Rubisco. Here the right option is option C. Due to the binding of O2 only, Rubisco affinity towards CO2 will decreases. Fixation will decreases. Due to what? Photorespiration happens. 125. When O2 binds to Rubisco, RUBP is converted to dash and dash. A. Phosphoglycerate, phosphoenol pyruvate. B. Phosphoglycerate. C. Phosphoenol pyruvate and then phosphoglycerate. D. Phosphoglycerate and phosphoglycerate. Here the right option is D. 126. In photorespiration, A. Sugar and ATP are formed. B. Sugar is formed but not ATP. C. ATP is formed but not sugar. D. Sugar and ATP are not formed. Here the right option is D. 127. Assertion: photorespiration doesn't occur in C4 plants. C. CO2 concentration at enzyme site is high in C4 plants. So choose the correct one. Here the correct option is option A. Factors affecting photosynthesis. 128. Photosynthesis is depend on A. Internal factors of plant, B. External factors of plant, and then C. Both A and B. Here the right option is both A and B. 129. How many of the following are factors affecting photosynthesis? Age of leaf, orientation of leaf, internal CO2 concentration, amount of chlorophyll, number of leaves. So here the right option is option D, all the 5. 130. Blackman's which law comes into effect when the several factors affect any biochemical process? A. Law of limiting components. No. B. Law of limiting factors. C. Law of limiting components. D. Law of limiting factor. Here the year is different, so The option last one is right, or not 1905, law of limiting factor. Here photosynthesis is controlled by factors which is in a least supply, that is the limiting factor which will be very least. Easy trick to remember, least wins always. Always whatever the least is there, no, that will be wins, and then which will be max, that is going to control. The fact that is least available will limit the rate of photosynthesis. Even if other factors are high, they cannot increase the rate, even though they are very least, but the maximum whatever it is there, no, it won't affect that one. 132 from light. The relationship between the incident light and then CO2 fixation rate at a higher light intensity is: A. Rate is constant with increasing intensity. B. Rate increases with increasing intensity. C. Rate decreases with increasing intensity. And then D. None of this. Here the right option is option A. 133. At lower light intensity, the rate of CO2 fixation is increasing with increasing in light intensity. 134. Light saturation occurs at a dash of full sunlight: 5%, 10%, 20% and 40%. Here B 10%. 135. Very big increase in light intensity cause increase in photosynthesis, decrease in photosynthesis, no change in photosynthesis rate, none of this. If the very big increase in light intensity happens, then decrease in photosynthesis happens. Carbon dioxide concentration. 135. Which of the following is true? CO2 is a major limiting factor for the photosynthesis in nature. B. Light is rarely a limiting factor for the photosynthesis. C. Both of these, and then D. None of this. Here the right option is C. 137. The concentration of CO2 beyond which the CO2 becomes damaging over longer period is 0.03%, 0.04%, 0.05% and 0.08%. Here the right option is C, 0.05%. 138. The CO2 fixation rate increases up to the CO2 concentration of 300 ppm parts per minute, 400 ppm, 500 ppm, 800 ppm. Here the right option is C, 500 ppm. 139. At low light condition, which of the groups respond positively to increase in CO2? C3, C4, both, none. Here the right option is none. 140. At high light condition, which of the group respond positively to increase in CO2? A. CO2, B. C4, both, and then none. Here the right option is C, both. It must be C3, not CO2. 141. C4 plants show saturation at CO2 concentration of A. 240 microliters micro per liter, 360, 450 and 540. Here the right option is B, 360. 142. C3 plants show saturation for carbon dioxide concentration at 450 micro per liter. 143. Assertion and reasoning. Greenhouse crops like tomato, bell pepper are grown in CO2 enriched atmosphere for higher yield. Reason: C4 plants respond to higher CO2 concentration by showing increased rate of photosynthesis. So here the right option is option C. Assertion is correct but reason is wrong. Temperature. 144. Which of the reaction is most sensitive to temperature? Here the right option is dark reaction. 145. Which group of plants has a higher temperature optimum? C4, C3, both, none. Here the right option is C4. C4 has the most high temperature optimum. 146. Which of the following is true? Tropical plants have higher temperature optimum than the temperature plants. Option B. Temperature plants have higher temperature optimum than the tropical plants. Option C. Temperature and tropical have equal temperature optimum. D. Polar regions plant have highest temperature optimum. Here the right option is option A. 147. Water stress cause A. Reduced CO2 availability, leaf wilting, reduced surface area of the leaf, and then all of this. Here the right option is all of this. 148. This is the graph. In the given graph, in which region will plant not respond to increase in CO2 concentration at the region of A? Question number 149. Which of the following correctly explains Blackman's law of limiting factor in photosynthesis? A. Rate of photosynthesis depends only on temperature. When several factors affect photosynthesis, the rate is controlled by the factor nearest its minimum value. Photosynthesis increases indefinitely with increase in light intensity. Carbon dioxide concentration doesn't affect the photosynthesis. Here A, C and then D are wrong and then B is right. 150 and then last question of today's session. At very high light intensity, the rate of photosynthesis may not increase further because chlorophyll becomes inactive, carbon dioxide becomes the limiting factor, temperature decreases, oxygen stops photosynthesis completely. So here the right option is option B. So for this chapter we discussed 150 questions and one of the best topic I can say, and you may expect much more questions from this topic. So as much as possible just revise the topics which are very important, like cell cycle and this one and whatever the next topic will be going to be discussed. Okay. Transport and mineral chapters were deleted from your NEET session or else for your NEET examination. That's what we are not discussing those two topics. Next topic will be about the respiration. So for today I am leaving this session. Bye bye.